Genes in the DNA sequence are interesting because they code for RNA and proteins that make you, you, and me, me. Computers are used to locate genes in the genome sequence by scanning the triplet codons, which determine amino acids consecutively with no overlaps in the 6 reading frames from 5' to 3', looking for distinguishing sequences such as a start codon, usually ATG, which indicates where to start translation, though ATG is also an internal codon. An in-frame stop codon, TAA, TAG, or TGA, which terminates translation many nucleotides away from the start codon, as proteins consist of multiple amino acids. And even as in eukaryotes with non-coding regions or introns, triplet codons that determine amino acids are read consecutively from the start codon, but only in the exon regions. A triplet may be interrupted by an intron, but there can be no in-frame stop codons in the stretch of DNA to be transcribed into RNA, that is, the open reading frame. A promoter upstream of the start codon indicates where transcription begins. Promoters in bacteria contain 2 short DNA sequences located at -10 base pairs and -35 base pairs upstream from the transcription start site. At the -10 position is the pripnol box TATAAT, and the -35 element typically consists of the sequence TTGACA. In eukaryotes, there are many different promoter elements, such as the Tata box, for example, TATAAA, about 25 bases upstream of the +1 start site, the CCAT box, for example, GGCCAATCT, about 80 base pairs upstream from the start site, and the GC box, for example, CCGCCCC, about 100 bases upstream of the start site. There must also be transcription terminator sequences downstream of the stop codon, such as a GC-rich region and a poly-A signal, AAT-AAA, with a CA and GT-rich region downstream. Intron signals are found in eukaryotic genes. The signal where the intron is removed commonly begins with the dinucleotide GT at its 5' end and ends with AG at its 3' end. An internal sequence such as TAC-TAAC, about 18 to 40 nucleotides upstream from the 3' end of an intron, also provides a potential bonding site for intron excision. Another region indicating that a region may be a gene is the CPG islands. These sequences with a high number of cytosine followed by guanine with a phosphodiester bond linking the cytosine to guanine helped regulate gene expression. Gene prediction can be improved by information on codon bias, as while different codons code for the same amino acid, some are more commonly found in genes of particular organisms. Gene prediction is also improved by looking for similarity to the genes or functional units of related species. Speciation involves mutation to DNA derived from a common ancestor, but genes and functional units show more similarity than non-functional areas. The slower mutation rates in functional areas than the rest of the genome is because mutations in these regions are more likely to negatively impact the organism than mutations in non-functional areas. While the sequence data allows us to predict the presence of putative genes in the DNA, whether the predicted gene is a functioning gene must be verified by experimental means such as the presence of its transcribed and translated products. Now that we have explored how to find genes in the DNA sequence, let us test our understanding of the concepts involved with a few questions. Question 1. How many potential open reading frames are present in a DNA sequence? A1, B3, C6, D more than 6. The answer is 6, as triplet codons determine the amino acids, and the code is read from the 5' to 3' end sequentially with no overlaps and there are 2 DNA strands. Question 2. How many open reading frames, no stop codons, are found in the segment of DNA indicated by the sequence below? The answer is 3. The top strand has 2 open reading frames. The single stop codon, reading 5' to 3', is highlighted in yellow. The bottom strand has 1 open reading frame, while the other 2 frames are closed, as seen by the presence of stop codons reading 5' to 3', also highlighted in yellow. Question 3. Which of the following nuclear pre-mRNA nucleotide sequences potentially contains an intron? Do pause the video while you read through the sequences. The answer is sequence E. It contains the conserved intron sequence, a 5' GU, a 3' AG, and a UAC-UAAC internal sequence, providing a potential bonding site for intron excision. Sequence D has a 5' GU and a 3' AG, but contains no internal A for the bonding site during intron excision. Question 4. Locating genes on DNA sequences may include looking for the A. Presence of a long stretch of codons between an AUG and a stop codon. B. Presence of CPG islands upstream of the start codon. C. Presence of regulatory sequences such as promoters upstream of the start codon and transcription terminating sequences downstream of the stop codon. D. All of the above. The answer is all of the above. Question 5. Transcription termination in eukaryotes depends on A. A palindromic sequence that can fold back into a hairpin structure. B. Recognition of an AAU-AAA sequence. C. Polyadenylation. D. All of the above. The answer is B. Recognition of an AAU-AAA sequence. A hairpin loop occurs in prokaryotes, whereas polyadenylation is a post-transcription process. I do hope this video has helped you understand how to look for genes in the DNA sequence. Upcoming will be a video looking into linking genes to their function or functions. Do subscribe to the channel if you haven't already, so you will be informed when new videos are uploaded. Thank you for supporting the channel.